Good morning dear students. In this video I'll explain what is DNA replication. The process by which a DNA makes a copy of itself is known as DNA replication. In other language we can say that the process by which the DNA makes an exact replica of itself is called DNA replication. Exact replica means the DNA makes a copy which is almost identical. DNA replication process is semi-conservative. I'll explain in later lectures. It occurs before cell division in the S phase or the synthesis phase. And it occurs before both types of cell division, mitosis and meiosis. So DNA replication is the process by which a DNA makes a copy, an exact copy or exact replica of itself. And DNA replication occurs prior to cell division, both types of cell division, mitosis and meiosis. And DNA replication process is semi-conservative. It occurs in 3 steps. First step is unzipping. In unzipping, an enzyme known as helicase breaks the hydrogen bonds between the DNA base pairs. You know that 2 strands of DNA are coiled with each other, and this coiling is mainly because the hydrogen bond between the base pairs adenine, thymine and guanine, cytosine of the 2 antiparallel strands. In unzipping phase, helicase unwinds the double helix structure to create a Y-shaped fork. Next stage is priming. A short piece of RNA, which is also called primer, binds to the starting point of the separated DNA strand. So DNA is double stranded and it is ladder like double helix structure. In unzipping stage, the 2 strands are separated from one another, and this happens by the presence of helicase enzyme, because helicase breaks the hydrogen bonds between the 2 antiparallel strands of DNA, and it breaks the DNA base pairs adenine, thymine and guanine, cytosine bonds and creates a Y-shaped fork. Next stage is priming, where a short piece of RNA, which is also known as primer, binds to the starting point of the separated DNA strand. The third stage is synthesis. Here in this phase, DNA polymerase, which is another enzyme, builds new complementary strand by adding free nucleotides that match the original strand. So in the third or synthesis phase, DNA polymerase enzyme builds new complementary strand of one separated strand, and another strand is formed and adding free nucleotides that match the original strand.